It may take about 10 seconds. Yes.
It says meeting is streaming live.
Yes, yes. It will appear in 5 seconds.
Yeah.
You may start.
It has started now. Yeah.
Good evening. Welcome to the third public lecture of the Vigyan Vidushi program. I am Bhaswati Mukherjee and I will be coordinating this lecture. Vigyan Vidushi is the first summer school organized by TIFR for women students who have completed their first year of MSc in physics. It is a 3-week-long program which started from 1st of June this year. And owing to the extraordinary circumstances, this year it is being conducted entirely online. A part of this program involves interaction of the participants with 6 eminent women physicists through a series of special lectures. We are very happy to have Dr. Prerna Sharma from the Indian Institute of Science, Bangalore, to deliver the third public lecture of the Vigyan Vidushi program. The title of the talk is Collective Behavior of Shape and Isotropic Colloids. This lecture is also being live streamed on YouTube to reach out to a wider audience. I will now briefly introduce Dr. Sharma. Dr. Sharma is an assistant professor and DBT Wellcome Trust India Alliance Fellow at the Department of Physics, Indian Institute of Science, Bangalore. She completed integrated masters and PhD in physics from TIFR, Mumbai in 2011. Thereafter, she was a postdoctoral fellow at Brandeis University until 2014. Her research interests lie in the areas of soft matter physics and biophysics. I now welcome Prerna and request her to deliver her talk. There will be a Q&A session at the end of the lecture. Those who are participating via Zoom can write their questions on the chat box and also raise their hands. I will be conveying the questions to Prerna. If time permits, we shall also be taking up selected questions from YouTube. So over to you, Prerna.
Thank you, Bhaswati. I will share my screen now. Just a sec. Just a second. You can see my PowerPoint.
Yes.
Yes. Yes. All right. So, hello everyone. Welcome to this special lecture. I'm going to talk to you about collective behavior of shape and isotropic colloids. So this lecture is aimed at young female students in science, and so it is targeted at that level of audience. And some of the results are from my lab and some of the results are from the community. So it's a colloquium style talk. So before I tell you about colloids and what they are, why are they interesting, and what are the kinds of questions that people think about in this field, I wanted to spend a few minutes talking to you about women in science, which is the context of this program. So, okay, yes. So before we get that going, I also wanted to acknowledge my group members who have contributed to some of the results in this talk. With that, I also wanted to emphasize that modern day science is actually a team effort. So typically you have a group of people working together on a given problem, and that's sort of the hallmark of scientific research in today's world. So science is more of a social endeavor in addition to all the science that goes into it. So somehow my slides are not changing. Yeah, okay.
You can try again, just go out and come back if you want.
Yeah. So So women in science, right? So the phenomena is that you have less than 30% of world's researchers that are women. And so this is significantly lower than the share of world's population that is female. So that's obviously 50%. And so you have a large gender gap in the number of women pursuing science. So, so other speakers in this series of special lectures will present to you the systematic understanding of this phenomena. So how come we have this underrepresentation of women in science in all possible levels? What causes it? What is being done to alleviate? Why should you worry about it? And there are a number of reasons why one should worry about it. The one that personally worries me is that I think of science as a tool to achieve growth and progress in every aspect of our society. So, in fact, rational and scientific temper, if present at widespread level in a society, can help in eliminating many societal evils. It can help in technological progress that can lead to better health and better life for each and every one of us. But if there is an underrepresentation of women in science, then you are effectively hindering the use of this tool and also the development of this tool. And so with these few words on the context of women in science, I would like to share with you my personal journey in the scientific world. So like most of you, I started out as an undergraduate student at St. Stephen's College, and I was enrolled in this physics honors course. So now, interestingly, I somehow landed up in this course. In high school, I was very much interested in chemistry and I dreamt of pursuing chemistry, but through a series of events, I landed up in a physics honors course. And so it is my teachers who really initiated and sustained my interest in this subject. So they were the ones who showed me how beautiful physics can be and that I should pursue it. And so after 3 years of this undergraduate studies, I was so much interested in physics that I wanted to continue studying it further. And I enrolled in a master's PhD program from TIFR. And this is where my thesis advisor and other mentors introduced me to the idea of curiosity, creativity coupled with scientific thought. And so once I had all of this training, the question was, am I ready to be an independent scientist? And most students at this kind of age are themselves not sure if that is the case. And so, like any other student, I spent time as a postdoctoral fellow in US to get this idea that can I supervise other research projects? Can I do independent research? And here the role of my mentor was really to help me get that confidence that I can be an independent scientist. So throughout this series of training, what I'm trying to emphasize is that there were many other people who contributed to my growth and professional development that wouldn't have been possible no matter how smart I was. And so if you find helpful mentors who can provide constructive feedback, who can provide decisive guidance, hold on to them. That would be my suggestion. And through this long growth, I eventually found a job as a professor at IIC Bangalore. And here the circle comes, life comes a full circle, right? Because now I have my own students with whom I engage in this process of learning and knowledge creation, which is actually interdependent. So throughout this cycle, you will eventually realize that whether it is teaching or it is research, it's always a two-way process. So students teach their teachers as much or more than teachers would teach their students. And so with this thought, I would now begin my main focus of the talk, which is to talk to you about colloids. And one last point I would like to emphasize is that oftentimes it's mentioned that, because of this underrepresentation of women in science, they don't have enough role models and things like that. And I agree that yes, there are less number of role models, but this is where peer-to-peer mentorship really comes into play. So you can learn a lot from your own classmates, from your own peer group, which may not be as underrepresented as at the next level. And so in addition to all of these people that I highlighted, keep your eyes and ears open to your own peers. So now coming back to the major agenda of this talk, which is to talk about colloids. So what are colloids? Colloids are materials or dispersions that you would find in your real life all the time. In fact, I think of it as matter that really matters. So you begin your day with a cup of milk, and you use cosmetics, you use inks to write, write on paper. And all of these are essentially dispersions of solid particles in a solvent. So for example, milk is a suspension of fat and protein droplets in water. Cosmetics are suspensions of oil droplets in water. And paints and inks are suspensions of dyes or pigments in some kind of viscous solvent. So what is so special about these particles or droplets is their size. So they are about a few microns in size. And what the key property that gets imparted as a result of this is that they perform Brownian motion. And thermal energy becomes the most important energy scale in this system, which ensures uniform composition. So essentially, thermal fluctuations cause these particles to move about in the solvent and ensure uniform composition. So why should you care about colloids in addition to the fact that you see them all around you? Is that they are important for a wide variety of applications, right from the biological world, where the blue color of bird wings comes about as a structural color. So if you have pigments that absorb certain wavelengths, that's one way to display color. But in these birds, there is no really a pigment as such. What is really happening is that you have these small 200 nanometer globules of keratin protein, which are arranged in a certain manner. And this kind of short range order of the material gives you a blue color when visible light interacts with it. Similarly, I talked to you about cosmetic creams, which are oil-water emulsions. So remember, oil and water do not mix. So if you just take a beaker of oil, add some water and shake it up, given enough time, the oil and water will phase separate. So to stabilize these uniform compositions of oil in water or water in oil, you add what are called surfactants. So surfactants are these chemically heterogeneous molecules which have hydrophilic head and a hydrophobic tail. And so essentially this dual nature of the molecule makes them sit at the interface of oil and water and lower the surface tension between oil and water, thereby stabilizing the emulsion. So if the surfactant was not there, then all the water droplets would coalesce and phase separate from oil. Now it turns out that instead of using surfactants, if you use colloidal particles, half of which is hydrophilic, half of which is hydrophobic, then they're going to again go sit at the interface and lower the surface tension. But now because you have these solid particles, the stability of this emulsion is way higher, because the coalescence between two water droplets is really opposed by these solid particles which have hardcore interactions. Similarly, you also use colloidal particles in the area of water filtration. So imagine you have a beaker of muddy water, and no matter how long you wait, it's going to remain muddy. So what you do is you add a protein, which is a drumstick seed protein, which is going to coagulate all the mud into larger particles. So instead of having single particles of micron size, you get hundreds of microns of size of aggregates, which will then sediment much faster. So from these examples, what you can see is that you really need to control the colloidal interactions for a given application. So for example, here you wanted to order the colloids in a certain way. Here you simply wanted to aggregate them, and so on. And so I'll spend some time talking to you about what are the interactions between colloidal particles, and then that will give you an idea how these interactions can be controlled. So from a fundamental science point of view, colloids are really cool because you can watch them with optical microscope. So a microscope in a high school laboratory is good enough to help you visualize or watch these particles. So here is a movie where each of this dot is a single silica sphere. And I'll play this movie and you can see that they jiggle around. So this incessant jiggling is coming out of Brownian motion. And you can see that I'm able to record it with a normal camera. So a normal 30 frames per second camera is good enough to record these movies. And a simple optical microscope is good enough to visualize them. So that's the power of the system, that it is very easily addressable and therefore analyzable. So chemists have really figured out how to control interactions between colloidal particles. And so here is the first interaction that you should worry about, which is van der Waals. So you know that two uncharged atoms will attract each other with a -1/r⁶ potential. And for two atoms, it is considered to be a weak interaction. But when you have two macroscopic bodies, you have to sum up interactions between all pairs of atoms in each of the bodies. So then what happens is that when you sum up the interactions between all pairs of atoms belonging to a given particle, you get an attractive force per unit area, which is very large for colloidal particles. So what that means is that if the van der Waals interactions dominates between the colloidal particles, you will just end up having these aggregates of particles. And this is what we call a colloidal flock. And this, being a very large aggregate, does not show significant Brownian motion. So the way to overcome this aggregation is to add charges on the surface of the particle. So now imagine that in addition to van der Waals, you have grafted some surface charges on the colloidal particle. And then what will happen is that you will get some kind of Coulomb potential between the particles, which can be very large, hundreds of kBt of repulsion. And this e⁻⁻r/λ is referring to what is called a screen Coulomb potential. So essentially, when you have these ionizable groups on the surface of the particle, you get a charged particle, but you get the counter ions, the plus of that group floating in the solution. So then these plus charges sort of screen out this plain Coulomb repulsion. So now I've shown you the source of attraction, which is a short range attraction driven by van der Waals interaction, and a long range Coulomb repulsion, which is coming out of the charges on the surface of the particles. Now, this kind of electrostatic stabilization works really well if your particles in water, because water has a very high dielectric constant, so that leads to dissociation of these ionizable groups on the particle surface. But if you have oil, then if the particles are in oil, then this electrostatic stabilization is not going to work because the ionizable groups will not dissociate. So you have to come up with a different way of stabilizing colloids. And the way to do that is to graft polymers on the surface of the particle. So you add these long chain hair-like objects on the particle, and these polymers, these chains like to fluctuate. So they're also Brownian in nature, and thermal fluctuations lead them to explore many different configurations. So if you have two such colloids that come together, then essentially these chains are going to entangle with each other, and that will lead to lowering in entropy. And since these are hard particle systems, this is not favorable. So you end up having some kind of effective repulsion. I will come back to this idea of entropic repulsion or attraction later on as well in the slides. So what is hard sphere interaction, right? So when John Dalton proposed his atomic theory of matter, he envisaged them as really hard wooden balls, and that's how he tried to illustrate it to them. But technically, hard sphere potential really means that when two particles touch, there is infinite amount of repulsion, and at finite separations, there is neither attraction nor repulsion. So for such a system, your internal energy is simply dependent on the thermal energy, and in the free energy, temperature really scales out. So then what this is showing you is that the phase with the lowest free energy has the highest entropy. So at equilibrium, you will find a phase which has the largest entropy. Now, hard sphere colloids are a model system to look at phase behavior of this system. And I will show you their phase diagram. And so this is the phase diagram. So temperature on the y-axis, volume fraction on the x-axis. So as we had discussed, for hard spheres, temperature really scales out. So the phase diagram is independent of temperature. It's only a function of volume fraction. So at low volume fraction, you would expect the particles to be randomly positioned. And so you expect a fluid-like phase. What happens is that above a critical volume fraction, which is about 0.55, you see that the equilibrium phase is a crystal. Now this is surprising. Remember I said that in hard sphere systems, the equilibrium phase is the one which has the highest entropy. And so in this volume fraction, crystal has the highest entropy. So how do you reconcile with your naive understanding of order and disorder and how they connect to entropy? So essentially the idea is that you can take a look at example of a volume fraction of 0.64. So at 0.64, what is going to happen is that if you produce a disordered packing of these particles, then all the particles will be touching each other and there will be no room for any particle to move. Whereas if at the same volume fraction, if you produce a crystal, then the particles on the lattice side will have enough room to jiggle around. So essentially crystal has higher entropy because even though it lost the configurational entropy, it gained the vibrational entropy or the translational entropy, right? And so, and in between here, you have this phase which is called glass, which is essentially what has happened is that a dense suspension of particles is really not equilibrated to a crystal, and you end up having a highly disordered amorphous system which has other characteristic properties associated with it. And I'll not go into the details as to what distinguishes glass from a fluid and so on, but let's continue for the moment. So this is how a colloidal crystal looks like under the microscope. So each of this dot is a micron size particle, and you can see that this frame is running to hundreds of particles. And so by having this colloidal crystal, you can really ask questions related to, for example, pre-melting here or defect dynamics that you couldn't have answered in the atomic system. So in the atomic system, you cannot really, someone's mic is unmute, yeah. So you cannot really figure out you cannot really image the atoms and watch their motion simultaneously in the way you can do it with colloidal crystals. Similarly, in the area of colloidal classes, a large number of advances have come about that are absolutely unreachable in the context of atomic classes, because here you can really watch particles move. And so, for example, this study showed that there are dynamical heterogeneities in the sense that there are areas within colloidal class where the particles are kind of caged. So they are basically just rattling around in a local area and then suddenly make a transition to another local cage. Another example of fundamental studies with colloids is that of gelation. So you can watch aggregation of colloids in real time. So this movie is basically showing this aggregation. So blue dots are single particles and red dots are particles which belong to large enough clusters. So I'll play this movie, and so this is a 3D microscope where you can really image the sample in 3 dimensions in real time and watch gelation happen. And from these fundamental studies, you can figure out, how good is your coagulating agent, what is the kinetics of gelation, is it suited to the particular application or not, and so on. So now I'll switch gears and talk to you about shape and isotropic colloids. So far what we have done is we have looked at colloidal particles, you have focused on the spherical geometry, we have drawn analogies with atomic systems. All that is good. But then if you change the shape of your building block, very different and new behavior is going to appear. And we'll choose the most shape anisotropic object, which is a rod. Okay, and so let us, just as we looked at the phase diagram of hard spheres, we are going to look at the phase diagram of hard rods. So when you have rods, you have this complexity of 2 degrees of freedom. Not only can the rods move, they can also rotate with respect to each other. So you have a translational and rotational degree of freedom associated with it. And so what that does is that you get what are called partially ordered phases. So at low volume fraction, you have an isotropic phase where all the rods are randomly pointing at each other. Whereas when you increase the concentration just a little bit, you get what is called a nematic phase. So what is a nematic phase? It's a partially ordered phase in the sense that all the rods are pointing along the same direction. So they're pointing along the same direction, but their center of masses are randomly positioned. So there is orientational order, but there is no translational order. So that's what I mean by partially ordered phases. And then if you increase the concentration further, you're going to get into this phase, which is called a symmetric phase. It is a layered phase of rods where each layer is more or less like a nematic, but then you have this 1D ordering happening along the z-axis. So these phases have interesting optical properties. So here what you're doing is that in the white light, the isotropic and nematic phases phase separate. So isotropic is a low density system and nematic is a high density phase. And so if you take a Waals which is in the coexistence regime, you visualize this separation. Now if you image the same cuvette under cross polarizers, what you will see is that isotropic will not give any light, whereas a nematic phase will. Why so? Because nematic phase has, now it's an anisotropic material. So it has 2 refractive indices, one along the rod axis or the directed axis and one perpendicular to it. So what happens is that when you pass linearly polarized light through a nematic phase, it gets rotated. Its plane of polarization gets rotated, and therefore you get some light on the other side. And then there is this idea of topological defects, that within the nematic phase, you will observe what are called topological defects, where the director is not well defined, and will not go into the details of defects here, but I just wanted to bring that up in your knowledge. So liquid crystals are interesting because of all these partially ordered phases they show, and they have a very large number of applications. So for example, you can get liquid crystalline molecules. So these molecules are essentially few nanometers in length, but they are really rod-like, right? And so they show these isotropic nematic transition as a function of temperature as well as electric fields. So in liquid crystal displays, switchable windows, essentially what you're doing is you're controlling the orientation of the rod like molecules, and thereby controlling the transmission of light. So for example, in a liquid crystal display, you have a backlight source, And then you have these molecules in a thin film. When you apply an electric field, then it orients them along the direction of the electric field, and then that controls how much light is allowed to pass through the. So that's how your displays work. So my lab also works on liquid crystals, and we work on rod-like particles, but we work on colloidal rods. So colloidal rods, and the particular colloidal rod that we are interested in is a FT virus, which is 1 micron in length, few nanometers in diameter. So interestingly, we are in this period where we are suffering from a viral epidemic, and this COVID virus the size. COVID is actually spherical in shape, and that size is about 100 nanometers. And so a COVID virus particle is also like a colloidal particle. The particular virus that my lab works on, which is called FD virus, it's a bacteriophage. It infects bacteria. It's non-pathogenic. And the beautiful thing about it is that it is as long as 1 micron and few nanometers in diameter. So the advantage of using viruses for our studies is that they're highly monodispersed. So virus is nothing but a piece of DNA coated with proteins. And so nature really engineers them to be highly monodispersed. So chemists, for the moment, are not very good at making rod-like particles with high monodispersity. And even if so, then the aspect ratio remains really a problem. And so because of all of these reasons, it's advantageous for us to use FD virus. And the key property of this virus that will lead to very complex phase behavior is what is called chirality. So if you zoom onto the surface of this virus, what you will find is that the proteins that are coating this virus are actually helically wrapped. So you can see that these proteins are kind of twisting around the long axis. And so this kind of substructure leads to what we call as chirality. So by chirality, what I mean is that two rods don't want to be perfectly parallel with respect to each other. They want to maintain a certain angle with respect to each other. So here in this graphic, what I'm showing is that the rods are twisting with respect to their neighbors. And so what that does to the bulk phase behavior is that you get what is called a cholesteric. So instead of getting a nematic phase, what you get is a cholesteric, where the director or the rod axis is rotating as you go along the z-axis, right? So if you observe such a material under cross polarizers, you get this stripe-like texture, which is a hallmark of a cholesteric phase. And so the reason these viruses are behaving as chiral objects is highly debated. So nobody really knows whether it is the fact that the proteins are helically wrapped that is leading to the chirality, or is it the charge distribution, or it's something else. And people know how to change it by adding dopants and things like that. But the general idea, why should hard rods or hard screws here should twist, is that you should think of them as screws essentially. So if you have two screws and you want to increase the free volume, or in other words, decrease excluded volume, you would want them to sort of intergroove with each other by coming at an angle. So, so essentially the behavior of these FD viruses is governed by a competition between an attractive interaction that I will introduce with the chirality. So basically, if I have many such viruses in the solution, they're just going to be isotropic and not do anything at low volume fractions. So what we do is we add what is called a non-adsorbing polymer, or think of them as small spheres which do not stick to these red rods. And what these small spheres will do is they will exert an osmotic pressure and push two rods together. Now this interaction is highly directional because you should think of these spheres getting excluded from the hard rods. So essentially, hard particles cannot interpenetrate each other. So this gray zone around the red rod essentially indicates the area within which the center of mass of the polymer coil cannot enter. So if you have two such rods that come close enough such that their gray zones overlap, then excluded volume will decrease and some free volume will become available for the polymer coils. So as we said, that phases with highest entropy will will be the phases of equilibrium. Tells you that the equilibrium structure for this will be for the rods to come together. So if you have many such rods, you will end up with a disc or a monolayer of aligned rods. So the gray zone overlap is maximum when the rods are aligned with respect to each other. So the phase with maximum entropy is the phase where rods are close to each other and aligned along the same axis. So if you have two such discs, they can stack up on top of each other through the same interaction. So essentially, now you should think of each of these discs as a colloidal particle undergoing this interaction with these non-adsorbing coils, what we call as depletion interaction. And so they stack up on top of each other. But we should remember that these objects are actually Brownian. So they can do thermal fluctuations, or thermal fluctuations are good enough to kick them around. So you get what are called protrusion fluctuations, which tends to move them up and down. So essentially it's some kind of random motion above the plane of the sheet. And so here you can visualize, so these green lines are individual rods, in red is your disc-like object, and you can see that they go a little bit up and down. And you can analyze it to find out that at high osmotic pressure, which means at large polymer concentration, the surface of these discs are very smooth, and so they stack up on top of each other. But at low polymer concentration, you have these significant protrusion fluctuations. And now if you try to bring two sheets on top of each other, you're going to suppress these protrusion fluctuations, which is entropically unfavorable. So you end up with these monolayers of aligned rods. This is how it looks under the microscope. So if you look at the sheet from the top, it will look like a circle. If you look at it from the edge, it will look like an ellipse. And so if you have two sheets, they can coalesce with each other and make a larger sheet. So you end up with these really large sheets, tens of microns in diameter, one rod length thick. So this is how colloidal monolayers look like, right? So here you are looking at these sheets from the top, tens of microns in diameter, and I've labeled 1 out of 10,000 rods. So these single dots are rods that are individual rods that are pointing at us, and they're freely moving around. So here is a sheet which has long wavelength elasticity but has fluid-like dynamics. So if you think of a standard plastic sheet, all the atoms are more or less at their position. But here you have a sheet which is fluid-like in nature, where the rods are really moving around within the sheet, but you can manipulate this sheet. The sheet has a finite elasticity associated with it, and I'll illustrate that with examples. So how does chirality, which I told you are the microscopic feature of the constituent rods, impact these monolayers? The way it impacts is that in the bulk, the rods are really standing straight. They're pointing at us, but at the edges, they lie in the plane. So essentially the edge of the colloidal monolayer is twisting. So chirality, or the chiral interactions, are expelled to the edges. And you can quantify this expulsion using polarization microscopy. And here the brightness of the pixel is proportional to the local tilt of the rods. So the rods are not tilted here in the center, they're tilted at the edges. And you can do some kind of 3D conoscopic microscopy to figure out which way the rods are twisting. It's a detail that I will skip here. You can take an intensity cut on this image to figure out what is called the twist penetration depth. So essentially intensity is low here, high here, and low again. And the way it decays from being high to low into the membrane gives you what is called twist penetration depth. And it's analogous to what London penetration depth. So there is a one-on-one analogy between how a symmetric phase expels chirality and how a superconductor expels magnetic field. And so this analogy was first elaborated by Dijen, for which he won the Nobel Prize as well. So yeah, so now why should you study monolayers? So I made an elaborate case on why you should have studied, for example, colloidal spheres, because the idea was that if you studied colloidal crystals, you could say a lot about atomic crystals. Now, the colloidal membranes are interesting because you can say a lot of things about cell membranes. So our cells are covered by this thin layer, thin two-dimensional sheet, which is made up of lipids. Lipids are again heterogeneous molecules with hydrophilic head and hydrophobic tail, and they spontaneously assemble into a bilayer to prevent exposure with water. And they're a few nanometers in thickness. So again, the problem is the real system is very thin and so very difficult to study experimentally. But the model system of colloidal monolayers is very easily visualizable, manipulatable, and can give insights into what's happening here. So how can it give insights about what's happening in a cell membrane? The kinds of questions you can answer are, for example, how do you shape a cell membrane? So many of the processes in cells are dependent on shaping the cell membrane. So for example, when cell eats something, it has to sort of make this kind of imagination and then chop it off, right? And this is how you can look at it under transmission electron microscope. So this kind of budding of cell membranes is a well-established phenomenon. But clearly, using a transmission electron microscope to visualize this process is very hard experimentally. And the fact that a cell membrane is actually a very complex system. It has hundreds of different kinds of lipids and proteins. So finding any systematic mechanism is extremely challenging, which continues to be a topic of active research. And a simple-minded physicist like me would say that let's stick to this toy model of colloidal membranes or colloidal monolayers, which are one component system. Another context where this analogy can be very helpful is in the context of phase separation. So what you're looking at are fluorescence microscopy images of real cell membranes where some kind of proteins and lipids have aggregated. So these aggregates have a lot of functional value, but people don't understand why these aggregates form, what stabilizes them, and so on. And so what is also known in all of these studies is that cholesterol is a crucial component. And cholesterol is actually a chiral molecule. So in other words, role of chirality in cell membranes is highly debated. And what I'm going to show you now is how we can use phase transitions and internal stresses driven by chirality to shape these model membranes. So I will spend another 5 to 10 minutes giving you examples and quickly going through them because I don't have much time left to give you a flavor of what we've been doing. So here you can visualize the edge of the colloidal membrane and you find that this edge fluctuates. So remember, all of these are Brownian objects. And so thermal fluctuations are good enough to cause them to move. And so by analyzing these fluctuations, you can calculate what is called the line tension. So remember, for 3D droplets, we were looking at surface tension. For these 2D droplets, we get what is called line tension. And now I'll show you an example of how you can control the line tension. So as I mentioned, these viruses are chiral in nature. And so here is the pitch of the cholesteric as a function of temperature. So essentially by increasing the temperature, you can make the rods achiral. So what that does is that at very high strengths of chirality, that is at low temperature, when chirality dominates, what's going to happen is that the edge is going to become unstable. So why so? Essentially, when you increase the strength of the chirality, it is favorable for the rods to be at the edge, because that's where they can satisfy their chiral interactions. So in this movie, I have a colloidal membrane whose temperature we will lower. And when you lower the temperature, chirality really dominates. And so, and depletion loses out. So essentially now the edge becomes unstable, and you get these ribbon-like objects where essentially these are linear objects where you can get lots of edge, and the rods can satisfy the chiral interaction. And this transition is completely reversible. If you go back to room temperature, it's going to come back to this high line tension state and become a circle, right? So that is one example of how chirality can control the line tension of colloidal membranes. Another example where chirality really introduces shape instabilities in the context of crystallization. So I had shown you these fluid colloidal membranes whose edges fluctuate, which do not have any positional order. So the question that my lab asked was very simple. So what happens when you increase osmotic pressure so that you get formation of a crystal? In other words, we were hoping to get this kind of a flat crystalline sheet, but what we actually found out was this, that the sheet was highly buckled and wrinkled. So the whole study was really trying to figure out how this instability comes about. And so I'll spend 2-3 minutes more talking about that, and then I will conclude. So, so essentially, first I need to really prove that this is crystalline. So if I want to prove that the sheet is crystalline, I need to do some kind of diffraction experiment. And because the length scales are very large, the diffraction angles become very small. And so we use what is called small angle X-ray scattering. And we found a well-defined peak for our solid membranes. So that showed us that yes, indeed, these are crystalline. And you can also look at the dynamics. So here you have a fluid sheet where all the rods, individual dots are moving around. If you look at the solid sheet, you will find that all the dots are static. So essentially the rods are completely frozen in, and you can characterize it quantitatively by measuring mean square displacement versus time. So for the fluid, it will look like a linear graph, whereas for the solid phase, it's a very small number and flat as a function of time. So if you want to really know what is going on, how you can go from a fluid phase to a solid phase, you really need to have another parameter. So to watch the kinetics, what we do is we take a polymer whose osmotic pressure is temperature dependent. So here I will induce crystallization by lowering the temperature. So what you can see is that a crystal sort of nucleated, and this crystal is spontaneously wrinkling. If you let it play on, then what you would find is that it will just grow and then cover up the entire sheet, and then the sheet can melt again if you increase the temperature. So here you're going to increase it again. And so you will come back to the fluid phase. So, so I've shown you both the transitions. So what is really going on? So what is really going on is the following, that essentially by crystallizing these chiral rods, you have really suppressed their chiral interactions. So this is a 2D simulation where the white means all the rods are aligned, and 0 means the rods have a finite angle. So if you evolve this kind of configuration under a energy Hamiltonian, which depends on the chirality, you will find that it becomes spontaneously unstable. So what I'm really trying to say is that a uniformly oriented state of all rods is unstable and will spontaneously form these grain boundaries where the chiral interactions are satisfied. So a single crystal of aligned rods becomes unstable to this formation of grain boundaries where chiral interactions can be satisfied. So this is sort of the crux of the story. And I will escape the PowerPoint mode and just go to the conclusion, which is to say that colloidal membranes are useful mimics of cell membranes, and chirality determines their phase behavior. And there was a third part that I didn't show, which was talking about active colloids and how activity changes the phase behavior. So thank you for your time, and I'm open to questions.
Thank you, Prerna. There are a few questions already, so maybe we'll take them up one by one. So, because I didn't want to interrupt you, may have to go back to your some of your slides. So, the first question is that is the nematic phase reason behind why some plastic materials give birefringence?
Could you please say that again? The voice broke. Is the
nematic phase reason behind why some plastic materials give birefringence?
Yes, exactly. So in plastics, essentially, you have these long chain polymer-like molecules. So you can also think of them as rod-like molecules. And when these plastic sheets are molded, these rods align due to flow. So it's called flow-induced alignment. And so in a cellophane sheet, all these molecules are aligned, and that gives you birefringence. Exactly. Yeah.
Okay. So the next question is, how or why does magnetic crystal rotate linearly polarized light?
Yeah. So right. I think it should be nematic. So I didn't discuss the optical properties of symmetric phase. So essentially, what I'm saying, so you should think of, this is more like a fundamental physics kind of thing, that so the nematic phase has these two directions, right? One along the axis where the rods are aligned, and one which is perpendicular to that. And the refractive index in these two directions is different. So imagine you come with a linearly polarized light that is at some finite angle with respect to these two axes. Then you can break it up into two components, and these two components will travel at a different speed. And so essentially it will come out at a slightly different angle, and therefore will get passed through the analyzer. Does that make sense? Hopefully it does.
Well, yeah, the person who asked this question should respond on chat. Okay. So there is one more question. Yeah, so she says she got it. So there's one more question. What is gelation?
So gelation, yeah, in a non-technical term, is essentially gluing of constituent particles. So in a colloidal dispersion, if it's a stable dispersion, all the particles are just moving about, and gelation will be like an aggregate of these particles. So when particles get stuck to each other and you have a system spanning network of these clusters, that's when you would say your system has completely gelled.
Okay, there's one question which maybe you will follow, but the question is, can you please explain again what the overlapping gray zones mean?
Yeah, I'm sorry, like, I went over it a little bit quickly. I can try to do this again. So, okay. So as I mentioned, we are thinking about hard particle systems. So what does hard particle mean? Hard particle means that two spheres cannot interpenetrate each other, or a sphere and a rod cannot interpenetrate. Okay. So now take this blue coil. So this blue coil will touch the red rod when its center is at the periphery of the gray zone. So essentially, gray zone is the area where if you try to put the particle's center of mass then the particle and rod will be penetrating each other. So this gray zone is the excluded volume zone. So what does it mean? Excluded means you are excluded from that area, or you're not allowed to enter that area. So essentially in this gray zone, you will not find a single blue particle. And now when two gray zones overlap, there is lesser gray area around for the particles, which means the particles entropy is going up. And the number of particles here is much more than the number of rods. So even though the entropy of rods is going down, the entropy of particles is compensating for it. So because the entropy of the particles is increasing, it's favorable for the rods to come together and have these gray zones overlap.
Okay, so the next question is so I think there is a question which reads as, how does the formation of crystals, how do the crystals probably become unstable when they form? I think that's probably the question.
Okay. I think it's related to...
Yes.
Yeah, so I think... it's related to here, right?
Yes.
So it's the crystal here. So essentially what I was saying is that imagine you make a crystal of chiral rods, right? Where the rods are actually just parallel to each other. So the rods are chiral, but you have forced them to be parallel with respect to each other. Now this is an energetically costly configuration. So under certain parameter regime or where chirality is very strong or dominates, what you would like to do is you would like to have some kind of compromise. So this is a compromise phase in the sense that this is still a single crystal, but now you have these grain boundaries where the rods are tilted. So by having these local areas where chirality is satisfied, you have this compromise phase coming up. Of course, if your depletion interaction or the interaction which began crystallization is very strong, then you will not get... Somebody is writing on my screen for some reason. Yeah, thank you. So then you will not get this kind of instability. So in the limit where the depletion interaction is very, very strong, you will not get this instability. And in the limit where chirality is very strong, you will not get crystallization. So in this competing regime, you will get this funny, frustrated phase. And experimentally, these grain boundaries show up as these wrinkles. So that was the point of showing you this simulation.
Okay, so the next question is, for colloidal glasses, is it due to the dynamical heterogeneity that the particles from one local cage are going to another cage, or is it due to random motion?
It is due to random motion. By dynamical heterogeneity, what is really meant by, let me go to that slide. So heterogeneity means things are not the same everywhere. That's the literal term. Dynamical means that things are not the same everywhere and they're changing with time. Okay, so essentially this cage-like motion is just simply coming out of the fact that it's a very dense system. But then you have these areas where there are areas where particles are just simply trapped in cages. And then there are areas where particles are more mobile. So that is the heterogeneity part, that there are areas where particles are more mobile versus areas where particles are not so mobile. And the fact that particle would jump from one cage to another is ultimately driven out of Brownian motion. But it's like a Brownian motion with lots of constraints, because particles around it have to move in order for this one to move to a different area and so on. Okay, so yeah, that's that.
Yeah, there is a somewhat exotic question which asks, is this mechanism also valid for rod-like structures such as axons?
Such as?
Axon.
Accents are like these muscle elements, no?
No, particles. These.
What are accents? I'm not aware of it.
Oh, okay, I think we can give it a pass.
Yeah.
So the next question is, what are the applications of anisotropic colloids?
What are the applications of anisotropic colloids? Yeah, so I showed you applications of anisotropic molecules. I did not show you applications of anisotropic colloids. It's a very good question. They are often used in what are these bulletproof jackets, and like where you would have some kind of material which would sort of become very rigid on impact and things like that. Also, if you couple it with magnetic if you have magnetic colloidal rods, then you can come up with all sorts of applications. But I don't have like off the hand very good application to show you.
Okay, the other question is, how much do the optical properties of pneumatic and cosmetic materials differ?
Yes. Differ. They differ in the sense like they will also, symmetric phases will also be birefringent. The where they will really differ is in terms of the kinds of defects they can have. So I did not talk about the topological defects in very great details. And so you will still get birefringence, but the kinds of topological defects you will get will be different.
Yeah. Okay. There's one question: can dyes have chirality?
Depends on the structure, right? Like dyes are typically ring-like aromatic compounds, like benzene kind of ring-like objects. So you really have to look at the structure to be able to say whether they are rod-like or not. Yeah.
Okay, I think there's one more question. It says that in your slide, colloidal crystals, glasses, and gels, you
Yes.
said the entropy is higher in crystal because it gains vibrational entropy, although its orientational entropy is gone. Then according to this point, the fluid phase should have maximum vibrational entropy. So what actually enables the crystal to have higher entropy?
Yeah, so the point is the following. The fluid has higher vibrational entropy at low volume fraction. At high volume fraction, crystals have higher vibrational entropy than fluids. So that's what I was referring to when I said look at 64% volume fraction. So at 64% volume fraction, which is an important number, it corresponds to what is called random close packing. So here, if you have a fluid phase, all the atoms or all the particles will be really jammed against each other. They will not be able to move at all. Whereas at the same volume fraction of 64%, if you put the particles in a crystalline geometry, you will have enough space to rattle around. So that's the argument. Here there is no orientational entropy. There is a configurational entropy and there is a vibrational entropy. In the rod-like system, we had orientational and translational entropy. So by configurational entropy means in how many ways you can arrange, in how many different ways you can arrange the particle. So of course, fluid will have very high configurational property compared to a lattice, because lattice is one regular arrangement in that set. But then at these high volume fractions, fluid is going to have lower vibrational entropy, and that's why the crystalline phase becomes stabilized in this hard sphere geometry.
Okay, so there is a clarification regarding the accents. So she meant like the neurons.
Neurons,
so,
yeah.
Yeah.
So that's a very good question. So neurons are like these filament-like objects, and any filament-like object in principle is susceptible to a partially ordered phase. But what sets the isotropic to nematic transition is dependent on the aspect ratio. So your aspect ratio has to be very high in order for you to see isotropic to nematic transition. I'm not aware of the literature of neurons, but certainly if you look at filaments like microtubules or actin, inside the cell membrane, there are also filamentous objects. And there people talk about, getting partially ordered phases and so on. So yeah, that's my short answer.
Okay, so I have a question for you. So when you are sort of creating these molecules in the lab,
Yes.
Do you see a preferential distribution of molecules of one chirality over the other?
Yeah, that's a very good question. So the nature.
Yeah, only one. Yeah, 100 things.
Only
Yeah.
one-handed. So the nature says, okay, it has to be right-handed, and all of the viruses are right-handed in that purification. But my lab has a library of these viruses. So essentially, by tuning the DNA of the virus, you can tune the chirality. And so we have both left-handed and right-handed viruses at our disposal. So we cannot get rid of the chirality, but we can make racemic mixtures and so on. So yeah.
Okay. Yeah, so I do not see any further.
Right.
Okay, so I think there is also a request to explain what is chirality by some simple example.
So take a look at a screw, and you will see that the thing around it is sort of going in one direction. Or you could think of like in the ball pen you have these little springs, and so these springs are again chiral objects. So I mean, there are many ways to talk about chirality, mirror asymmetry, and things like that. I don't want to go there because, just think of it as a spring, and then each spring has this, helix direction, clockwise, left-handed or right-handed, and that's it. That's your chiral interactions. I mean, chirality right there for you.
Okay, so I think you have more or less answered all the questions. That was a wonderful talk, and I'm sure you have inspired the young students. I have known you as a student, and it's really my pleasure to host you today. And known you as a very enthusiastic and a very sincere student, and so I'm very happy to see you as a PI and doing so well. Congratulations on that. And so I think with that, on behalf of the organizers of Vigyan Vidushi, I once again thank you for joining us here and sharing some of your work and life with the participants. I'm sure they would derive a lot of inspiration from that. Thanks a lot.
Thank you.
So on that note, we end this public lecture.
Yes.
And thank you, students, for joining in. Thank you so much.
It was my honor. Thank you.
Should I stop live?